Thank you very much, Tanya, for this invitation, and we'll go round. Maybe Charlotte, you can enter the fray.
I'm Charlotte Acharya, and I work with Richard Michelmore at UC Davis for doing COVID testing, and then joined the variant analysis team. So I'm a staff researcher.
So I'm Richard Michelmore. I'm director of the Genome Center, and I oversee the operation, but of course, all the hard work is done by the team.
I think I might actually introduce myself similarly, actually. So I'm Tanya Nolan, and I've recently joined LGC to head up their assay design and development team. Maybe we could think back for a few moments and kind of set the scene for how we have ended up where we're at and how the collaboration formed. I was thinking back to the beginning of the pandemic over a year ago now, and how frustrated I was that I wasn't really able to contribute and make use of my molecular biology background, because I was one of the early people to actually catch COVID, which meant I was isolated for several weeks. So as I was thinking about that, and I was thinking it'd be really interesting to hear from you, Richard, how you came to be in the position of doing the COVID testing at UC Davis.
Yeah, well, somewhere there's some parallels, and the Genome Center is a high complexity lab in the true sense of the word. Mm-hmm. We do some very sophisticated molecular biology. And in March, April, when the COVID was really blowing up, there was clearly a tremendous need for testing, and the medical infrastructure was not able to handle it. At the same time, I knew what was possible in the ag biotech sector. Mm-hmm. I knew the ag biotech sector had the capability of generating millions of data points a day. But then really LGC and us got together to implement the high throughput testing system. And of course, once we got that up and running, and we've now done over 500,000 tests, wow. The technology, of course, really was set up, designed to detect SNPs. Yep. So it was a very small conceptual leap to say we should be doing high throughput testing to complement sequencing to monitor variants.
Absolutely. So when did you get the testing up and running?
I campaigned for about 2 months to get the funding. And then it became very obvious, it was inevitable that they were going to have to do testing if they were going to open the campus in any shape or form. So the provost gave me an email saying, okay, here's the green light. And it was like, oh, what have I got myself into? It was becoming apparent that saliva was a perfectly good medium. Mm-hmm. And we also, the saliva workflows that people were talking about was 2 or 3 mils into large tubes.
Wow.
And, and if you're only using less than, a microliter in your reaction, you don't need that. Mm-hmm. So we went with a very small volume in barcoded tubes, and the barcoded tubes were put in 96 racks in the collection kiosk.
Yeah.
Right. So much simpler workflow than anybody else was envisioning. But the problem is saliva is viscous.
So this viscosity problem, we could lean on protocols developed at University of Illinois and Yale, but in our first tests with our setup, with a super high throughput setup that we're using for the LGC equipment and 380 channel pipetting reactions, we still had problems with that. We tried proteases testing, and I thought it was worth trying to try papain.
Yep.
And that was, at least to my knowledge, a new approach for the saliva issue problem. So papain turns out to be working really well, and that's what we're using now routinely all the time. And papain has the purpose in the papaya plant to actually attack the digestive system and necessarily also mucosa from insects that are trying to eat
Right.
on the trying to feast on the papaya.
Uh-huh.
That's its biological purpose, and that's why we assumed it would really be potentially worth trying.
Yeah.
Because, you know, our mucosa and saliva, these are most likely related proteins, enzymes. It works very well. Yeah. So this is our, after the heat inactivation of the sample, this is our first step. And the processing, making saliva samples really pipettable without any problems.
As you reflect back, having achieved this tremendous objective, if you were now going to think about giving advice to anybody else, maybe as we're starting to see issues arising in other countries where maybe they don't have the same infrastructure, is there anything that, as you reflect back, you will give as advice?
Well, actually, the molecular biology is relatively straightforward. What shouldn't be underestimated is the front end and the back end. So, you know, we've talked about what happens in the lab, but actually these sample collection kiosks and things like that. But one of the big decision points was, did we have one centralized kiosk? Did we have a dozen kiosks around? Did we do some form of home collection? And it transpired, I think, we initially started with 12, but we ended up with one giant one. It was so much more efficient. And we've got now indoors, we've got a testing facility that spans full basketball courts, but it's very fast, it's very simple. Although it might be a little less convenient for people to actually go to one location, when they get there, it takes them less than, well, 2 to 4 minutes to go right the way through the system. And we have a lot of control. So the QC on, we can say we need this amount of saliva in the, you know, so, but it was an enormous operation. You know, there's a blue and a gold team because it runs 7 days a week. And there's about 60 people associated with the kiosk. The high throughput system with the IntelliCube that we got from LGC, that is a very simple machine. I mean, it does so much of the work for you. And the team isn't all that big. So we end up with 3 teams. We had a blue and a gold team that were kind of the production teams. And in the initial phases of the epidemic, they were not meant to interact because heaven forbid that one of them gets infected and therefore you have to quarantine. So we really tried to keep 2 production teams separate. And then we had the development team, which was led by Charlotte, Vanessa, and Lutz. So the idea was they were the troubleshooters, and they could step in if one of the production teams went down, but it didn't. And then, of course, testing doesn't solve the problem, right? It's a tool, and therefore you've got to have a good contact tracing. So the medical, you know, interfacing with the medical. And one of the things that we have is that we report back the results very fast. We're reporting back, I think, close to 40% of the results the same day and 95% of the results by the end of the next day. So that allows very fast contact tracing. But again, then you have the bioinformatics, and the UgenTech plugin turned out to be a very good strategic decision for us. The CLSs really like it. It helps because if you're doing thousands of samples a day, you've got to be able to handle the data and, again, result them very, very fast.
That's the absolute benefit, isn't it, of having a streamlined testing and reporting and isolation, and that's how we'll control the virus. Well, that kind of, I think, talks a lot about the initial testing. So then I came into the story probably shortly after Christmas, when we then started to become aware. Well, we've been aware, haven't we, since last June, I think. We, as in Stephen Buston and I, published the D614G, so we were already becoming aware of how mutations were arising and how they were potentially impacting. And so then we started to have conversations around different ways of tracking variants or tracking mutations to then define variants. What are your recollections of that time?
Too far ago. I can't remember.
5 months, I know, it's a lifetime.
Again, it was slightly that the amount of sequencing being done was woefully inadequate. So sequencing is very important for discovering variants. And this is when the B117 that was first identified in the UK was beginning to become prevalent. And that was actually identified because of effectively a SNP, which was the S gene dropout, that the part of the multiplex assay for the S gene wasn't amplifying because of the deletion. So essentially, the discovery of B117 was a SNP assay. Yeah. Inadvertently.
And a failed assay, but yeah.
It was a failed assay, but basically it was SNP. And so again, I got a little frustrated with and I actually, to some extent, still remain. There is a tremendous pressure to sequence more and more for monitoring of variants. And this is really taking a sledgehammer to the problem. And I think tens, if not hundreds of millions of pounds and dollars are going to be wasted by misapplication of technology. If you try and sequence everything, the amount of information you get back as you sequence more and more of the same thing, asymptotically, goes like this. I mean, you're getting very little extra information when you sequence to 100,000th B117. As we said, the IntelliCube and the Nexar technology was set up to analyze SNPs in very high throughput. So the technology was there to do the analysis if we had the assays. And it was really a logical next step. And we've now put it in place. And we look at 100% of our positives get SNP genotyped, 100%. And then if there's anything interesting, we then sequence them. Right. So it's a complementary technology.
Well, I think that's the key, isn't it? That we can see how, if you already have a testing workflow, then the variants, the way that we have them up and running, can slot into that very easily. Maybe we should take a minute to explain what we've done to measure those variants. So really the approach has been, as you say, that classical genotyping, because that's the background, but recognizing that as the virus is mutating, there are specific mutations that can identify those emerging variants. And by testing those SNPs, then we can define the variant. So each of the variant assays has got the 2 generic primers and then 2 probes that have a different label, one for the wild type. By wild type, we mean the original Wuhan sequence, and then one for the mutation, which in many cases is a single base. We've got some deletions in there. We've got some multiple nucleotide polymorphisms as well. Of course, we're reliant on sequencing to get that mutation information to begin with. So it's absolutely critical there. But in terms of monitoring populations and looking at prevalence and spread of different variants, as you point out, the real-time system is just so much faster. Endpoint. So if you think about your own sample that you did at 1:30 and you got your result at 9:00, if by chance there was a positive, how soon do you think you'd be able to, using the variant assays, define which variant we were looking at? How quick is it for you?
It depends slightly. Well, when we had a fair number of positives, we were running the assays twice a week. Right. So within 4 days. Now the problem is if you only have one positive, it's like, I think last week we only ran all the assays once. Right. But if we really did this in high throughput, we could be doing this every other day. We thought we would be doing it every other day. So When we've got, when we first got samples of B1, B1351 or B117,
Yeah.
we were knowing within 3 or 4 days of sample collection that we had a variant, and we were then informing the health authorities of that. So the assay itself, I mean, it's a day's work.
Yeah.
It's really when connecting, getting enough samples, in fact, to run the assays versus, because it is a high throughput machine, you're putting it on the IntelliCube. You need a fair number of samples to make it do it.
Yeah.
It's a nice problem to have.
If we recognize that some of these mutations have impacts in terms of transmissibility, or even now when we're starting, we've got really successful vaccination programs, but then we're also saying, well, are there any of these mutations that are going to protect the virus against immune responses, which as an early adopter I feel really irritated about. But then, even tracking these mutations just so that we then know how effective the vaccination program is going to be and when we need to think about boosters. So when you're talking about being able to get a result back in even a couple of days, surely that's got to be of tremendous value to people making decisions about how to contain the virus.
Now, one thing that's important to understand here, though, is that this is not an FDA-approved test.
Absolutely.
And coming from my background and your background, I have to say that working with plants is a lot easier than working with human samples, because for regulatory constraints and things like that. So
absolutely.
we can apply this information for public health purposes.
Right.
We cannot report it back to the health provider.
No.
And the result cannot be reported back to the individual.
Absolutely.
And we've got to be very careful about that.
Yes.
So it can go to the public health authorities and they can make decisions based on the information we give them as to whether it's a new variant. And you touched on, we're very aware of vaccine breakthroughs, vaccine escapes.
Yeah.
And of course, whenever we get one of them, we immediately genotype and sequence it anyway.
Yep.
There is going to come a time, almost certainly, unfortunately, as an evolutionary geneticist, the selection pressure is so steep for the virus to evolve so that vaccines are no longer effective.
Yeah.
And I think we could almost be back to square one. And you're going to get an upswing again. And this monitoring, though, is going to be absolutely critical.
Yep.
But the challenge is going to be that if an individual walks in where we know that strain is much more virulent and the vaccines are not effective, that is probably actionable with regard to that individual. But we can't
do that.
We can't report that back at the moment. And so, this is something we're discussing. And how do you actually validate a test? Because we would need to validate the test. And how do you validate it? Well, you validate it by sequencing. But then actually getting clear certification for sequencing is in itself challenging.
Absolutely.
So
how do we know the sequences are right?
Exactly, and that's not actually a trivial question. So I'm on work groups to grapple with this question, but it's not an easy one. What's great about the Pfizer and the Moderna vaccines is that it is a relatively short development cycle.
Yeah.
And yes, they can deploy different RNAs. So yes, you're going to need to have a booster shot. But how do you deploy the booster shot? And whether there's the political will to ramp up the testing again, to do the masking, do the social distancing.
Yeah.
I mean, it's feasible in places like the UK and the US to do this.
I'd like to take us back a little bit to thinking about the development of these assays and some of that iterative process, and also take the opportunity to bring Lindy in, because Lindy
right.
was the person who did a lot of the actual coalface of those designs, and maybe for us to think a lot about what some of our starting assumptions were and some of the things that we learned through that process. I know certainly as we were doing them, probably one of the things I would reflect on is how there were several times that we needed to abandon standard software, and I had to kind of rely on, Lindy calls it my Boy Scout approach, but it's actually my orange marker pen, and very much relying on your own experience, I suppose, and saying, well, the software won't give us anything suitable, so I'm afraid it has to be the orange marker pen solution. So I know for some of the assays that we did do that. But Lindy, I'd like to invite you to think about the learning process that you've gone through, and if you were now going to advise somebody else about how to design assays for these variants, knowing what you know, what kind of things might you share?
Yeah. When Richard first approached us, it was over actually the Christmas holidays, because I remember working on them. He was like, do you have anything for N501Y? We need to track down this UK variant. And I was ready for him. I didn't know Richard at the time, but I was ready for someone to ask me that question, because it had been in the news a couple weeks prior. And in my, at the bottom of my to-do list, since no one had actually asked me to do it. But there we go. We were off to the races, and I started with our general process for designing a BHQ plus SNP assay. But what we found right away was that we needed to make the assays much shorter than what we typically go up to 150 bases to find suitable primer sets. But with the RNA degradation, even with the papain, we were still experiencing some challenges with sample type. So we brought in the assays to closer to what the diagnostic assays were running, between 65 and 75 base pairs. But as we were developing them, it seemed almost at a weekly basis at this point, there would be a new mutation. And the areas where it was mutating, they were hot spots. So we would have to incorporate either shifting the assays to try to avoid the new mutations, or use degenerative bases to try to accommodate them and keep the assays working. And it was really Richard's team who tested, we still don't have the right answer, of which there isn't for every target, there isn't a prescribed way to do it. It still takes taking an informed approach, but it's really the team in Davis that, put these assays through their paces and their feedback that really guided to what they have now, which, correct me if I'm wrong, Richard and Charlotte, but you're working with a panel of 12 assays that you use in your production runs for lineage definition.
Yeah, it's as you said that, I had a recollection as well of one time when we were working on, I think it was the 681, and we had an assay, and Richard and Charlotte had tested it. We were really happy with it, and we were just about to get another synthesis done, and a mutation arose. And we've not even got through the testing of this yet. And there's another mutation already. We've got to change the assay, not because it's not working, but because we needed to make accommodations. Yep. So absolutely.
And another interesting thing with these variants is there is a convergence in their evolution. The strategies that are the mutations that arise that permit the virus to be more infective don't happen just once. So we're 5-6 months down the road now. There's assays that were designed in February that really weren't that popular, but now they're coming back and they're just as important and informative. And so with having this selection of different targets, different SNPs to target, we're really getting to a place, I think, where we can give information about specific lineages and ones that haven't even arisen yet. I was seeing this week the C36 lineage, which is that the Taiwanese lineage?
Yes.
It's
variant
it's
under
carrying the
investigation.
677 mutation, which is one months ago we hum and hawed about of, well, we've got the assay, it's working really well, but we don't think there's a market for it. But now there might be.
It could be, absolutely.
Well, and also the Vietnam variant that the press is getting, saying it's a hybrid, but really it's not. It's the 617.2 plus the 144 deletion.
Right. Yeah.
Which
So it's the same idea.
It's an example of convergent evolution.
Absolutely.
But again, we've got all the SNP assays to detect it.
Yep. So by using this system, you can mix and match that panel, as you referred to, the production panel, and we can drop them in and out. Absolutely. Yep. And I suppose the other final point, unless other people would like to contribute to the topic, is the other area that, of course, we've been monitoring all the time, because while we've been designing assays to detect variants, we've also had to have the ongoing screening at the other end to make sure that the detection assay also remains effective. And so far, we're confident that it is doing, but we do know that there are many detection assays that are experiencing problems for exactly those, the reasons we've just talked about. Some of these mutations are unpredictable, some of them are reasonably predictable. Absolutely. As we were thinking back, I really was aware of what an incredible journey it's been. And I think there are some nuggets of gold in there to share with people who are in areas where they don't have the privileges that we have. I mean, even though you had to fight with the university to get money, at least there was money there available eventually.
Right. And I will say, but once the trigger was pulled, everybody really got behind it. It was remarkable. Yeah. And the very top administration says, make it happen. And everybody made it happen. We ordered the IntelliCube in 3 days. I mean, bang, it just went through everything. So, the university's been tremendously supportive. But your point about global deployment, in many areas, there are ag biotech companies with this technology, right? And really, all you need is a conventional PCR machine with a fluorescent plate reader.
Absolutely. Yep.
And it won't be perfect, but it'll be better than you'll be able to. So it's a very low tech. Unfortunately, I think this story has still quite a ways to run.
I have a friend in Ecuador, and of course, all of their focus is on genotyping or genetically characterizing the different species of whatever the endemic creatures are. But of course, they've turned all their attention towards testing, and we're talking about pretty low throughput facilities, really. But even up in Esmeraldas, which is a lot of that area, they don't really have ready access to healthcare, but they have real-time instruments. And
exactly. So getting the supplies out there, and I think one of the things that we can do is to get the WHO on board.
Yes.
If we can get WHO to say this is the technology that needs to be put globally, because we need to solve the COVID problem globally, not country by country.
Oh, absolutely. When we look at the PHE reports that I know we both look at, that tells us huge numbers of the different variants are certainly coming into the UK because of people traveling, and people are always going to travel. So the virus
that's
is
what
just
we
going to
see
mix
here, I
around.
think, from basically from the variant analysis, we're getting the COVID we're seeing is migration in.
Yes.
So we get these variants are coming in, and they don't blow up. They die out. But it's because it's continual migration in.
Yeah.
We're seeing the little COVID that we've got.
Right. Yeah. Interesting. Fantastic. Thank you.
Anyway, thank you
so
also,
much.
Thank you guys. It's been a great and very productive interaction, and we need to keep doing it.
I think so. I think there's a really, really, really important message here. You know, as we think about the comparison of time and cost of sequencing, it just doesn't make sense to do anything different. The genotyping is just so fast and cheap, isn't it? So.